Hello and welcome to this A-level biology revision video on enzymes inhibitors. Right, in exams, this is often an area that people overlook when they're carrying out the revision. But there are usually 1 or 2 marks available in the exam for it, and within most enzyme questions there will be some or a small section on enzyme inhibitors. An enzyme inhibitor is a molecule that binds to an enzyme and decreases its activity. Since blocking an enzyme's activity can kill a pathogen or the correct metabolic imbalance, many drugs are enzyme inhibitors. They are also used in pesticides. Inhibitor binding is either reversible or irreversible. Enzyme inhibitors reduce the rate of an enzyme-catalyzed reaction by interfering with the enzyme in some way. This effect may be permanent or temporary. So inhibitors can either be competitive, in which case they will compete for the active site, non-competitive, in which case they will bind to a second site on the enzyme and change the shape of the active site, or uncooperative, in which case once they're bound to an active site they will not unbind from it, therefore deactivating or denaturing, if you wish, the enzyme. Competitive enzyme inhibitors work by preventing the formation of an enzyme-substrate complex because they have a similar shape to the substrate molecule. So they block the active site. This means that they fit into the active site but remain unreacted since they have a different structure to the substance. Therefore, less substrate molecules can bind to the enzymes, so the reaction rate decreases. Competitive inhibition is usually temporary and the inhibitor eventually leaves the enzyme. This means that the level of inhibition depends on the relative concentrations of substrate and inhibitor, since they are competing for places in the active sites. So the graph below shows the rate of reaction with a competitive inhibitor and without. So eventually, with a competitive inhibitor, you will still reach the maximum rate of reaction. However, it will take a lot longer. Non-competitive enzyme inhibitors work not by preventing the formation of enzyme-substrate complexes, but by preventing the formation of enzyme-product complexes. So they prevent the substrate from reacting to form the product. Usually, non-competitive inhibitors bind to a site other than the active site called an allosteric site. Doing so distorts the 3D tertiary structure of the enzyme so that it can no longer catalyze a reaction. Since they do not compete with the substrate molecules, non-competitive inhibitors are not affected by substrate concentration. Many non-competitive inhibitors are irreversible and permanent and effectively denature the enzyme which they inhibit. However, there are a lot of non-permanent and reversible non-competitive inhibitors which are vital for controlling metabolic functions in an organism. Enzyme inhibitors by organisms are used in controlling metabolic reactions. This allows product to be produced in a very specific way and in very specific amounts. Enzymes vastly increase the rate of a metabolic reaction, often by a factor of up to 10 million. This fact is essential for all life on Earth, but it means that enzyme activity must be tightly controlled since uncontrolled reactions can be fatal. Uncontrolled reactions might also lead to an increase in metabolic activity, which might cause the temperature of an organism to rise too high. It might cause all of an organism's stored nutrients to be used up too quickly, so they'll end up dying due to lack of nutrients. And there are a wide range of other systems that could be disrupted if metabolic control wasn't undertaken. Often a metabolic process is composed of many different reactions, each of which is catalyzed by a different enzyme. These reactions are called metabolic pathways. For example, photosynthesis has a metabolic pathway. In many cases, the final product of a metabolic pathway acts as the non-competitive inhibitor to one of the enzymes earlier along the chain. This means that the metabolic process controls itself, since more product gets produced, the more it inhibits the pathway and the slower the process proceeds. In this case, that would be an example of negative feedback where the product is controlling the activity of the enzyme, in this case, that is producing that product. Right, I hope that this video has been helpful. Make sure that you go online, find some questions to do with enzyme inhibitors and use of enzymes. Good luck in your biology exam.